All right, so today we're going to talk about how parasites or pathogens survive. So obviously, every time you get an infection, your immune system doesn't always clear it, and sometimes you get sick. So let's see what the pathogens can do to avoid your immune system. So one of the first parts of your immune system that we've talked about is phagocytosis, macrophages and neutrophils coming through and eating up foreign objects. So pathogens have ways to prevent or stop this eating. And so this figure from your textbook is just explaining pathogens can potentially release toxins that kill the phagocyte. They can prevent contact so that the phagocyte cannot eat them. If they do get into the phagocyte, some pathogens have different methods to not be digested, right? So they might block fusion with the lysosome, which has the digestive juices. They might be able to escape. They might produce something again that prevents the killing by the lysosomes. And this chart, and there's a whole, this is a much longer list if you go to that reference, but I just put up a couple illustrations to show you that some pathogens have multiple ways to resist phagocytosis. Again, killing the phagocyte, inhibiting chemotaxis, so they might produce something that doesn't even allow the macrophage or the neutrophil to be recruited, avoiding detection, resisting the killing, and here's the ones that inhibit the lysosome and the fusion of the lysosome. These I'll show you are some intracellular parasites that actually live inside your immune system cells. Another way to evade your immune system is blocking the cilia. So remember, the cilia is lining your trachea. It's going to your lungs, and it has all these little hair-like projections that help brush things away. So a good example of this is the bacterium Bordetella. What am I doing? Bordetella pertussis. I hope I spelled that right. This is what causes whooping cough. And you do get a pertussis vaccine in that Tdap that we talked about last time. But this bacteria actually produces a toxin that can paralyze the cilia. So now the cilia cannot sweep things out of the trachea. Bordetella also produces fimbriae. Do you remember this word? Fimbriae. I think I spelled it right. That allows it to adhere to the cilia. So now it has a nice place to colonize and grow and cause this. That's the whooping cough sound when people have that type of infection. It causes inflammation. It just causes a hard time to breathe. As adults, we usually can get over it, but it's really hard on children. Other things, just to FYI, cigarette smoke, pollution, cystic fibrosis, These things also affect the movement of the cilia. Cigarette smoke and pollution can destroy your cilia, so that's why people who smoke have what we call smoker's cough, because they don't have these nice cilia to help move things out. Cystic fibrosis is a genetic disease where there's a huge accumulation of mucus, so the cilia can't work to clear things out, and then people with cystic fibrosis can get infections in their lungs because they can't clear out the bacteria or viruses. Another part of the innate immune system that we talked about is complement. So different organisms have different ways to interfere with complement. So remember, a couple ways that complement works is to tag the microbe, to poke holes in it, to tag it for opsonization, which means phagocytosis. And so there's just generally different strategies to interfere with the regulators, degrade the regulators of complement, bind them up so they don't work. Some bacteria actually will use complement to trick the cell into taking it in, and then they become an intracellular parasite. Another thing that pathogens can do is they can disrupt signaling. So you might block interferon signaling, so the cells are not alerted that there is a viral infection. You can have something called a super antigen, which is kind of like a decoy. It says, okay, T cells and B cells, here's something you need to make lots of antibodies or lots of cytotoxic T cells to. And it's really just a waste of resources because it's not an antigen that will actually target the pathogen. Another interesting example is Epstein-Barr virus, which replicates in B cells. Epstein-Barr will actually turn on B cells and tell them to make a whole bunch because that's the host that it needs. So it is disrupting the normal signaling and saying, make a whole bunch of these B cells so that I have plenty of cells to infect. This is a table from your book. I apologize, they don't give me the tables, so I have to try to scan them in. Immunosuppression means you're knocking down, you're suppressing the immune system. And the examples here are affecting the B and T cells. Okay, so we're at the adaptive immune response. Okay. So some organisms actually infect immune cells. So like HIV infects T cells. Right? Epstein-Barr infects B cells. And so on. And so some of these pathogens suppress your immune system by infecting those cells and either killing them or disrupting their cytokine production. And so these temporary ones are really important because it gives the organism, the pathogen, time to replicate and spread. And just so you know, CMI stands for cell-mediated immunity. So we're talking again about that adaptive immune response and affecting the cells important for that. Here are some intracellular pathogens. Okay, so these actually hide out inside your cells and live there. And so they're not targeted by the immune system because they're in your own cells. So the immune system can't tell that these cells are infected. Viruses, some viruses do this, and some can produce what we call a latent infection. So a latent infection here means that there's no virus being produced. But the viral DNA is there, or RNA. So it's just kind of hanging out, kind of hibernating. Then you get some kind of reactivation event where now cells produce virus. Okay. You get some disease, and then it usually shuts down and goes back to latency. So a great example of this that many of us know about, is herpes, right? So every time you get a cold sore, you have a reactivation. The times that you don't have a cold sore, the virus is in the latent stage. It's hard for the immune system to deal with this. It can only work here where you're producing virus. It shuts it down, but the virus genetic information is still hanging out. Okay. HIV is similar. HIV actually integrates into the genome, so it can just hang out there and replicate, and when it's activated it makes virus, and when it's inactivated it just hangs out there with the genetic material to make more virus. Persistent infections by definition produce a little bit of virus or bacteria. Okay, this is what we also call chronic, right? So your long-lasting infection, you're producing a little bit, not enough to get a huge immune response, and these can lead to, like it says here, chronic disease, right? So hepatitis B and hepatitis C is a great example. They can lead to liver cancer and liver damage because they're always there producing just a little bit of virus to mess up the cells. Now you can see herpes is on here, as well as HIV, cytomegalovirus, Epstein-Barr, varicella. So there's a very fine line between persistent and latent, and many viruses go between both categories, whether they're producing some pathogen or whether they're in kind of hibernation. Another way for pathogens to evade the immune response is to replicate in what we call privileged sites. So this is parts of your body where there is not much immune surveillance. Okay, not many B and T cells. So this is an example of virus. This is human papillomavirus. And it's replicating, you can see, in the skin as it differentiates. And so we don't have the antibodies and the T cells circulating there, so this virus can replicate undetected. You can evade the immune system by shedding to the surface, which, remember, shed is like exit. Okay. So again, you're in an area without much immune surveillance. So remember that your B cells and T cells are constantly going around through your blood, through some of the tissues. Macrophages are going there looking for foreign objects or things that are identical to the B cell or T cell receptors. And so if you're shedding in a place where maybe you just have to deal with antibody, such as a gland or a duct, then you're going to be able to evade that massive immune response and produce much more pathogen. Evade the immune response, form your own protection. So this is a cyst, and in that cyst is tapeworms. Okay, you can kind of see that little guy right there. And these tapeworms make these fluid-filled sacs where the worm can live. This happens to be from somebody's lungs. And it's just this nice protective coat that the immune system cannot penetrate. Look like self. Okay. This idea is limited to some helminths. This is, I didn't write it down. Schizosoma, hold on, I know I wrote it down over here. Should I spell it right? S-C-H-I, It's horrible when you get old and you can't read from far away. Schizosoma, okay, which is a blood fluke type of helminth. And this guy actually goes and coats itself with glycolipids from your blood cells. So the things that make you have either type A or type B blood, it will coat itself with that, right? And so then your immune system doesn't recognize it as foreign. Or it can coat it with MHC, another part of your immune system, cell signaling, coat it with MHC antigens. So again, your body doesn't recognize it as foreign. Another trick that some viruses and bacteria do is they produce what we call an FC receptor. So if you remember our structure of antibodies, these are the variable regions and this is the constant, and this is called the FC area. And remember that the variable is where it specifically binds the antigen, right? So this is where specificity comes in. So this diagram here is showing you a microbe that has made this FC receptor so that it's actually binding the antibodies backwards. So now the antibodies can't go and attack it, right, like it normally would, to tag it and then signal an immune cell. So they're basically kind of sequestering or sucking up binding up all the antibodies so that the antibodies cannot tag them. Couple more strategies. One is change what you look like. Okay, so change the outside, right? And remember that that's what our body is making the adaptive immune response to, is what's on the outside. Make T cells that can recognize it, make B cells that can recognize it. And so if you're constantly changing what the outside looks like, you have to start a new immune response every time that parasite looks different. So this is really common in trypanosomes. Okay, so these are protozoa. And this is actually called gene switching. So even within a single host, they can turn on different genes so that they look different as they're replicating and making more parasites. So the poor host that's infected in trypanosomes can cause African sleeping sickness. This poor host is working against this variant, knocking it down, and all of a sudden another one comes in. And so it's not because it's been reinfected, it's because the pathogen has changed its gene expression, changed what it looks like outside within that single infection. A organism that's really good at doing this is influenza. Maybe I shouldn't call it an organism. Remember, this is a virus. And influenza has two different strategies. Antigenic drift is mutation. So these are small changes. Okay. HIV does this a lot, which is why we haven't been able to make a vaccine against HIV, and why we have to have a new flu shot every year, because these viruses are constantly changing what they look like on the outside. Now, influenza can also do something called antigenic shift, and this is large changes. So see how they're trying to illustrate, you go from a purple triangle to a purple square, and here you have totally different combination. And what influenza does here is a single cell is co-infected by two totally different influenza viruses. So they've color-coded the genomes here. And what happens is you get reassortment. So influenza has 8 RNA molecules that make up its genome. And so if a cell is co-infected by two different viruses, then it can recombine that genetic material, and in this case it got 4 from one and 4 from another, and now look like a totally different virus. And finally, some pathogens just do what we call hit and run. They invade, they multiply, and they leave. Right? Before you even know what hit you, you're sick, and then a lot of times you're recovering really quick. So things like rhinovirus, which is the common cold, head cold, rotavirus, which is a gastrointestinal system, 24-hour flu, right? Hit and run. Make a whole bunch, get out, try to find the next host, before you even get the adaptive immune response. So this is one of the reasons you can keep getting these head colds over and over again, or you can get the stomach flu over and over again, because you haven't even had a chance to get your adaptive immune response up and running before the pathogen's gone. So those are some of the strategies that interact with our immune system that we talked about last week, and how the pathogens can avoid that or modulate or control the immune response. And so your last lecture will be about pathology, and really what we're talking about is how our overactive immune response can cause damage to our own tissues. All right, thanks.